Welcome everyone, and thank you for joining today's webinar titled 3 Techniques, 1 System, How to Effectively Characterize Complete Muscle Function. My name is Andy Henton from Inside Scientific, and I will be your host for today's event. Our session today is sponsored by Aurora Scientific and will be focused on methodology, best practices, and strategy for implementing in vivo, in situ, and in vitro muscle experimentation techniques. First, we will hear from Matthew Borkowski, Sales and Support Manager at Aurora Scientific, who is going to provide a brief history and overview of Aurora and their design and engineering approach as it relates to muscle experimentation. In particular, he will introduce their 1300A 3-in-1 system, a combined solution permitting in vivo, in situ, and in vitro muscle tests. Following, we will be joined by Drs. Ramsey Carella and Christopher Ward of Myologica, a CRO that specializes in neuromuscular function and muscle phenotyping services. Together, they are going to review assay techniques and discuss how these muscle experiments can be used individually or together to effectively study muscle function for animal phenotyping.
Thanks everyone for joining again. Thank you, Andy, as I just said, and welcome. We're all very thrilled that you were able to attend the presentation today about the 3-in-1 test system. And today my fellow presenters and I will try to give you an overview of 3 techniques that we feel are very powerful that can be properly used to assess muscle function in your animal models. Now, before I begin giving you an overview of the system and the actual techniques themselves, I'd like to take a moment to just give you a bit of background about Aurora Scientific and my fellow presenters. So Aurora was actually founded in 1982 by 2 professors from the University of Toronto, and it initially spent its early years as a small custom engineering and instrumentation firm, and we've worked on projects as diverse as blood rheology, gas dispersion, microseismics, you name it. And it wasn't until 1997 that we began getting involved in muscle and muscle research. Initially, we only sold single instruments, force transducers and positioning motors, and in that time we've developed stimulators, software, experimental apparatus, and bath chambers, and our focus now is really packaging these instruments into systems and providing solutions that are as turnkey as possible for tests that range from single cell all the way up to the whole animal. And the 3-in-1 system we'll be describing today falls somewhere near the larger end of that scale. I'd also like to take a quick moment here to thank my fellow presenters, Dr. Ramsey Carella and Dr. Chris Ward of Loyola University and the University of Maryland, respectively. Both Ramsey and Chris are also the co-founders of Myological, which is a CRO that specializes in animal model phenotyping, compound screening, experimental design, and training in these techniques. Both have been longtime users of our instruments and are very experienced with these techniques that we will be presenting. So before I continue on to discuss our 3-in-1 system and the experimental techniques associated with them, I'm going to just introduce for you in a moment here a few other assays which may be more familiar to some of you in attendance. And these are the 3 that are pictured here. So the techniques shown on this slide, the treadmill, the running wheel, the grip strength, have been widely used for many years to provide a broad picture of an animal's physical performance. And while it certainly provides us with a piece of the puzzle, on its own, they don't actually tell us much about true neuromuscular strength of any individual muscle itself. We can't necessarily infer from the fact that an animal ran further or held on to a grip meter longer than another animal that this occurred because their limb muscles produced more force relative to their size than the other animal we're comparing against. So one of the main reasons that we can't make this inference is because of all these assays shown, grip strength and treadmill and wheel running, they all have a significant behavioral component. So did the mice not run as far because this model just simply isn't as active as another? Did we have an intervention to induce hypertrophy and in so doing make it uncomfortable for the mice to run because of their larger physique? These behavioral questions make a direct correlation between these physical performance assays and neuromuscular strength a bit murky. So to see the whole picture and to validate or disprove our hypothesis sort of about the holistic physical performance of the animal model, we require assays that specifically test how strong the muscles are relative to their cross-sectional area or the masses of the animals themselves. And we potentially even need to go beyond that to test the strength of the individual muscle building blocks themselves. So in so doing, we also need to remove the voluntary component of the assay entirely by activating the muscle directly with an electrical stimulus in this case, and not relying on the CNS of the animal model to sort of drive its peripheral nervous system. So I now present to you our 3-in-1 system. And so what is it, and what experiments can it perform? The first experiment is what we term in vitro, and is sort of pictured here on the right. And in this case, we remove a muscle of interest from the mouse or other animal and place it into an oxygenated bath chamber, which keeps it viable. We stimulate the muscle with field electrodes, and the force produced by that one muscle can be measured along with any simultaneous changes we make to muscle length. The term ex vivo has also been occasionally used for this type of experiment. The next experiment is similar in that we are still isolating a single muscle. However, there is we greatly reduce the amount of surgery that's necessary. We term this in situ, and it's performed by making a small incision in the animal's leg to expose one of the muscle tendons of interest. The tendon can then be cut and sutured to our system, and so thusly it's similar in that sense to in vitro. However, we have the advantage of not having to remove the muscle from the body. There's no bath to keep the muscle viable. We use the anesthetized animal itself. Its own blood supply is keeping the muscle viable. So the third and final experiment is what we call in vivo, and to some it's perhaps the least well-known experiment of the three. Although actually it's been described in the literature for over 25 years at this point, I'd say. So the experiment involves anesthetizing the animal and measuring the aggregate torque produced by either the entire plantar or dorsiflexors of the lower limb. There's no surgery required, and the attachment point, rather than a muscle tendon, is now a small boot, which is pictured here on the left, where we tape an animal's foot into. The contraction is produced by a small needle electrode, and the experiment is intended to be non-terminal. So how does it all work? Now that we know a little bit about what the experiments are, we can take a closer look at the engineering behind it. So the system is based around our flagship instrument, the dual mode lever, and that basically means that the lever system is not only a force transducer but also a position transducer as well. So in addition to being able to measure force and position, it can control either one of these parameters while simultaneously measuring the other, and it can flip instantaneously between the two. Really, the big thing is that since we only need one attachment point now to the muscle, This is really what enables being able to do some very sophisticated experiments in situ or in vivo without the sort of the one attachment point functionality. We sort of have no way to tape into a boot without using a separate motor or strain gauge system. And because it's not simply a force transducer, we're capable of performing far more complex protocols than simply isometric force or torque. And this allows us to really better provide a model of what the muscle is doing in real life. So just a few quick notes here about the rest of the system. We have an experimental apparatus that goes along with it. It's sized for the correct animal model in question, be it a mouse, a rat, or something much larger. We have all the positioning and accessories is built in with the actual apparatus to make it as turnkey as possible, and we allow for quick switching between the three different techniques. So if you want to convert between in vivo and in situ, it literally only takes about 2 minutes. As well, we have a dedicated software suite that we've written to specifically for muscle type of experiments. And we call this dynamic muscle control. All the stimulation parameters, any stretches of the muscle, any changes to isotonic tension, everything is fully controlled through the software. And because the package is specifically written for muscle experiments, we already have a library of standard protocols that allows for any sort of customization that you might want. We also like to think it's quite straightforward, keeping in with our philosophy in general about making the system as turnkey as possible. So once you've loaded a pre-canned protocol and the muscle or the animal is attached on the platform, press start and begin collecting your data. As well, there is a dedicated analysis package that we've written, and as we all know, sometimes it can take longer to analyze the data than actually collecting it. So we have written an automatic high throughput module for standard muscle type of experiments, and these script routines that run on the many contractions that you'll collect can typically analyze several days worth of experiments in a couple minutes. As well, we support the software quite heavily, and we have a team of dedicated programmers that are always looking to either add functionality or tackle a bug if you discover it. So that being said, as I mentioned earlier, we have a very diverse group of users of this particular system. People ranging from muscle physiologists and pathologists to bioengineers to neuroscientists, really anyone I think that has any kind of peripheral interest in muscle or muscle functionality could benefit from these techniques. And actually, our next presenter, Ramsey, will probably tell you a little bit more about that. So Ramsey, I leave it to you.
Thank you, Matt. Hello, everyone. I'm Ramsey Kerala, co-founder and president of Myologica, and currently also at the Loyola University Chicago, studying cardiac and skeletal neurofilament function. I'll be giving a nice overview of the three main types of experiments that the 3-in-1 system can perform: the in vivo, the in situ, and the in vitro measurements of muscle function. And I will start with the most physiological assay, in my opinion, the in vivo hind limb assay. An in vivo experiment allows the researcher to measure torque produced by the ankle or ankle torsion of one of two hind limb muscle groups. The first muscle group are the ankle flexors or dorsiflexors, which represent the TA or tibialis anterior, and the extensor digitorum longus, or EDL. This muscle group is responsible for flexing the paw of the animal towards its body. The second group are the ankle extensors or plantar flexors. This group is composed of the gastrocnemius or gastroc and the soleus, and is responsible for extending the paw of the animal away from its body. Other muscles can also be evaluated with some custom-made modification. Dr. Rich Lovering at the University of Maryland School of Medicine has developed a method to evaluate the quadricep muscle as it extends the leg around the knee. The same approach can be used to measure other muscle groups in the forelimbs or in the jaw. The main concept behind the in vivo ankle torsion assay is that it can be performed without surgical isolation of the muscle, disruption of blood flow, or alterations in the innervation. The assay can be performed in a mouse or a rat that is deeply anesthetized, usually via inhaled anesthetics delivered by nose cone. In the previous slide, you saw a mouse undergoing the assay, and here you see a rat. The animal is placed supine on a thermostatically controlled plate to keep body temperature stable, and the hind limb is mechanically isolated from the rest of the body at the knee. This is usually achieved by inserting a pin into the tibial head on one side and pressing the knee against a rubber dowel on the other side. This ensures the limbs does not move either vertically or laterally during muscle stimulation. We always ensure that the tibia is perpendicular to the foot lever, the hips of the animal are slightly raised, and the thigh is bent in relation to the knee. The foot is then secured in a foot plate mounted to the torque sensor. This foot plate has a groove that fits the ankle of the animal and ensures that the torsion is occurring centered on the axis of rotation of the torque sensor. The most technically challenging part of this assay is the placement of the percutaneous or subcutaneous stimulation electrodes to elicit contraction of the muscle. The stimulation of different nerves determines which muscle group is stimulated. The fibural or common peroneal nerve for dorsiflexion, TA and EDL, and the sciatic nerve for plantar flexion, gastroc and soleus. The electrodes seen in this pictures are placed for plantar flexion. With enough practice, the user can place these electrodes with very little variability between preparations and use as little current as necessary to ensure a maximal contraction. Following placement of the electrodes, current is increased sequentially and a single contraction elicited at each increase to find the current needed to elicit supramaximal stimulation. It's important to note that this assay does not directly measure force, but rather torque about a joint, in this case the ankle. Torque, also known as moment or moment of force, is defined as the tendency of a force to rotate an object about an axis. Just as force is a push or a pull, a torque can be thought of as twist to an object. Mathematically, torque is defined as the cross product between the lever arm distance and the force vector, which tends to produce rotation. In the ankle torsion in vivo assay, the foot plate acts as the lever arm, and the length of the lever arm is the size of the foot. To accurately report force, therefore, the torque measured by the sensor is divided by the length of the lever arm. The Aurora 3-in-1 software already performs a conversion using standard sets of assumptions, including the length and weight of the lever arm. Additionally, to account for differences in animal or muscle size, the force should be normalized to one of 4 measures. The most accurate normalization would be to muscle fiber cross-sectional area measured by histology. However, this method would require the muscle to be removed and would be a terminal procedure, thereby negating the advantages of the in vivo measurement. Similarly, estimates of muscle mass would require euthanasia. If possible, direct measure of muscle cross-sectional area by MRI and CT scan are of great value, although they are usually expensive and time-consuming, making them less suitable for large, higher throughput studies. Fortunately, in our experience, we have found that normalizing to animal weight, in most cases, is a very good surrogate for muscle size. So what are the pros and cons of the in vivo ankle torsion assay? Well, of the assays possible with the 3-in-1, the in vivo measure provides the most high throughput assay. With practice, one animal can require as little as 10 minutes for a few simple measurements. In addition, it is the most physiologically relevant assay as the muscle is kept in its native position, the assay is run across the joint, and the blood supply and innervation is kept intact. This further enables the measure to be performed serially over days or weeks to follow progression of a treatment or a disease, or recovery from an injury. However, some drawbacks must be kept in mind. The assay measures torque from a group of muscle, like the EDL and the TA, or the gastroc and the soleus. So a determination of muscle-specific contribution is challenging. Furthermore, this assay is dependent on the native innervation of the muscle to elicit a contraction. As such, any disease or treatment that affects the motor neuron or the neuromuscular junction would potentially underestimate the muscle-specific force. Finally, this assay is potentially the most variable, as electrode placement requires practice to achieve consistency and can be technically challenging. The second assay possible with the 3-in-1 system is the in-situ, or intact muscle measurement. It is a hybrid of the in vivo assay that I just described and the in vitro assay that I'll be describing later. An in situ experiment allows the researcher to isolate a muscle of interest but keep one or both ends of the muscle in situ so adequate blood flow is maintained. Similar to the in vivo assay, the in situ assay can measure force from the TA, the EDL, the gastroc, or the soleus. However, this assay measures individual muscles instead of entire muscle groups. Furthermore, any linear muscle that has a side that can be ligated can have its function measured with this in situ approach. This includes the trapezius, the diaphragm, the masseter. As this is an invasive technique, it is usually considered a terminal experiment. Once again, the animal is placed either supine or prone, depending on the muscle assay, on a temperature-regulated platform, and anesthesia is maintained via nose cone. The foot is taped down, the leg is pinned down, and the skin is retracted at least halfway up the muscle, and the muscle is surgically dissected from the adjacent muscle. The distal tendon is severed and tied to the force transducer lever arm with a silk suture seen here. The muscle can be stimulated 2 ways, either directly using platinum electrodes placed on the muscle or via nerve stimulation similar to the in vivo assay. The muscle is kept moist, usually with a saline drip or mineral oil to prevent drying. Again, similar to the in vivo assay, force must be normalized to account for differences in animal size. However, in this assay, given that the muscle is already dissected out, direct measure of muscle cross-sectional area is possible. Ideally, histology should be performed and fiber cross-sectional area should be used for normalization. This technique is definitely more time-consuming. Once again, muscle mass is proven an adequate surrogate for cross-sectional area, and yields an acceptable normalization parameter. In addition to standard assays, the in-situ preparation also allows to perform different ones, one in particular being the motor unit number estimation. A muscle is innervated by a single neural trunk that encompasses several motor neurons. These motor neurons branch and terminate at one to several muscle fibers. Stimulation of a single motor neuron would then elicit a contraction from one to several myocytes it innervates. To determine motor unit number, the investigator would prepare the muscle for an in-situ measurement. The neural trunk is then isolated and severed and suctioned into a simulation electrode. The nerve is then electrically stimulated with small steps in voltage, and discrete quantile increases in force are monitored. Using predictive modeling, the motor unit number is then estimated. So what are the pros and cons of the in-situ approach? Like the in vivo approach, the neurovascular supply of the muscle is left intact. This allows the assaying of larger muscles like the TA and the gastroc that is not possible using an in vitro approach. The surgical isolation also allows direct measurement of a single muscle's contribution to force, as opposed to the in vivo approach that measures contribution from a whole muscle group. Most importantly, the in situ approach is suitable for a hybrid nerve muscle stimulation protocol, allowing the investigator to functionally differentiate between motor unit stimulation and direct muscle depolarization. Those advantages do not come without a drawback. Surgically, isolating the muscle and severing the tendon is very invasive and technically difficult. Furthermore, because of the surgical preparation, temporal repeated measures are almost impossible. Finally, the last application possible with the 3-in-1 is the isolated muscle experiment, which is the classical experiment the majority of physiologists are familiar with, whereby the muscle is surgically removed and placed in an in vitro bath. This is the experiment, as opposed to traditional preparations where the muscle is hung vertically. The in vitro configuration of the 3-in-1 system places the isolated muscle in a horizontal, temperature-controlled, oxygenated chamber. The muscle is attached to a fixed post at one end and to the dual-mode lever at the other end. The dual-mode lever allows you to measure and control both force and length with just one attachment point. Complete characterization of the muscle is possible with only one attachment point, so a range of parameters including eccentric, isotonic, fatigue, and length velocity are easily tested. The bath can also be outfitted with perfusion, allowing the buffer in the muscle in which the muscle is bathed to be replaced and recirculated. This is particularly attractive if you're testing different pharmacological compounds or different doses of a drug. The muscle is stimulated using field electrodes, which are placed on either side of the muscle, or if the nerve trunk is left intact, using a suction electrode, as seen in this picture. In this image, we can see that the setup is using field stimulation instead, with the electrodes flanking the entire length of the muscle. Keep in mind that in this preparation, we remove the blood supply of the muscle. Therefore, the buffer must provide all nutrients and oxygen necessary for proper function. As such, the in vitro preparation is suitable for only a certain subset of muscles. Only small muscles whose metabolic need can be met through diffusion will survive and function properly. These include the EDL, the soleus, the lumbricalis, the diaphragm, and certain cardiac preparation like papillary and trabeculae. Muscles from a young rat can be suitable, but as the animal grows, the muscle size might start becoming limiting. This is obviously the biggest drawback of the in vivo approach. However, this method also represents several great strengths. Because the muscle is removed from the leg, the in vitro approach allows direct measure of muscle contractility independent of neuronal integrity or other variables like restricted blood supply. In addition, dissection of the muscle with a portion of the motor nerve allows ex vivo nerve muscle function. What I believe is the most attractive possibility allowed by the in vitro bath is that chemical or pharmacological manipulation is possible and independent from oral bioavailability or circulating dosage of your drug of interest. In fact, serial additions of a drug can help the investigator determine the optimal dose for a functional effect. Again, this technique requires some practice in order to perform the surgery, and temporal measures are impossible.
I'm excited to be here. The 3-in-1 system is in routine use in our laboratory. The in-situ, in-vivo, and in-vitro approaches described by Ramsey and Matt, really they've been long established and they're really foundational measures in neuromuscular physiology. With the 1300A, the 3-in-1 system provides is a streamlined platform enabling these approaches. And really each approach enables really a wide spectrum of assays. Here I'm just going to talk about a couple that the audience is actually likely probably familiar with, especially based on the polling. The first is general contractile function. So we'll talk in general about some isometric contractions and some concentric contraction type experiments. We'll look a little bit at fatigability, how to that we can fatigue the muscle within each of these paradigms. And of interest to the muscular dystrophy community and growing interest in many other communities is susceptibility to injury. And we'll show some examples of both eccentric and isometric contractions. So really underpinning much of this physiology are some basic assumptions. And as you get into this field, you realize that in setting up this equipment, the equipment has been provided. It's state-of-the-art, where it's important to realize that we need to make some basic assumptions and some equipment and animal setup to get optimal data. The first is we assume, based on our data, that we're collecting our data at optimal muscle length. And so that's the resting length of that muscle. That has to be set for each preparation, and that is a potential limitation. If we set it and know it, we limit variability. Also is the stimulation paradigm. What we try to maximize is that we provide supermaximal stimulation such that we're ensuring the recruitment of all the muscle fibers with even a single action potential. Indeed, that's not how the muscle works physiologically as driven by central stimulation. We have motor unit recruitment, but providing supermaximal stimulation decreases one of the free parameters that allows us to really get very precise investigation of our model system. And please here note that both the resting length and stimulation intensity are determined for each preparation by the investigator. There are a variety of variables that could be examined to gain insight into muscle function with all of these techniques. If we elicit a muscle contraction, we could examine the peak amount of force that's produced, the peak torque, or the peak force. We can also examine the kinetics of the contraction profile, and we'll introduce some of those. The most familiar assessment that the audience has seen are isometric contractions. These are the most common functional assessment. This is when a force is produced by a muscle with no overall change in the length of the muscle. So the muscle is, the overall muscle length doesn't shorten, however, force is produced. And so the classic way to investigate isometric contractions is by examining the force versus stimulation frequency relationship. This is when we have trains of action potentials that are delivered at increasing frequency until there is no further force increased. And as you see here, in a 250 millisecond train, or a 250 millisecond window of time, we can either deliver one action potential to deliver a single electrical excitation period that generates a twitch contraction, and as we increase the frequency of stimulation during that paradigm, we deliver more and more pulses during that period, we get an increase in the force that's produced until we get a maximal force. And so we can examine the peak force response for each stimulation frequency. We can also examine kinetic responses at each frequency. This relationship was explored in a recently published study by the Wagner Laboratory at Kennedy Krieger Institute and Johns Hopkins University. This was a collaborative study that my lab was fortunate to participate in. Here, the Wagner Laboratory examined wild-type mice, and these were compared to genetically modified mice. You see here the isometric force on the y-axis and the stimulation frequency on the x-axis. On the y-axis, the force was normalized to the calculated cross-sectional area of the muscle. This was alluded to as one of the normalizations by Ramsey earlier. And this is expressed as grams of force per centimeter squared of muscle area, of muscle cross-sectional area. And in this graph here, we determined that there was no significant differences between the genotypes in this study. That wasn't the end of the story. We had a hypothesis that, based on this genetically modified animal, that the contractile kinetics might also be altered. And so one of the benefits of the DMA software, the analysis end of the software, is that it provides the ability to analyze the kinetic properties of the data. And here we specifically examine the rate of force production over time, the dF dt positive, which is the rate of rise of the contraction, and the negative dF dt, which is the rate of relaxation of that contractile kinetic, as seen here. When this data was interrogated, you see that over the same stimulation frequencies, from 0 to 300 hertz, we saw a significant difference in the rate of rise of that contraction, but no significant difference in the rate of decay. And so that gave us really further insight into this genetic modification of this animal. And if you would care for more detail, the references provided at the bottom. This is just to highlight the fact that a thorough investigation of your data can yield quite exciting results. Those were in vitro experiments. Those were experiments where muscle was taken out of the animal, and that was an extensor digitorum longus preparation. In situ experiments are also very flexible and powerful. This can be readily seen in experiments done by our colleague Richard Lovering and recently published. And this is an in situ preparation. In this preparation, again, if you remember back in Ramsey's slides a couple slides ago, the animal is deeply anesthetized. Here, the muscle is surgically isolated at the tendon. Blood flow is maintained, and that tendon is attached to the force transducer. So in this experiment, what you see is a fatigue protocol. And so the initial pulses over time, what you see on the y-axis, these are individual force transients, and on the x-axis is time. And so every second for 2 minutes, a tetanic stimulation is being delivered, and we're looking at the decrement in force over time. And you see periodically, every 15 seconds, we switch from a nerve stimulation directly through the motor nerve to a stimulation of the muscle with electrodes placed in near proximity to the muscle belly. And so what you notice here in this experiment is that over time, this stimulation paradigm results in a significant fatigue when stimulated through the nerve that is not nearly as significant when seen by direct muscle stimulation. So here, you're able to dissect out the differences between phenotypic loss of force during fatigue between a nerve or a muscle stimulation. Functional assessments are critical to understand physiology in the neuromuscular axis. What is given is that in the intact animal, the muscles of locomotion transmit force across a joint, resulting in a change in muscle length and articulation of that joint. In the first two examples I gave, those are isometric contractions, so the muscle was not allowed to shorten. If we allowed the muscle to shorten, we have concentric contractions, where muscles actively shorten. Alternatively, we have eccentric contractions. These are active contractions, but during that contraction cycle, the muscle is lengthened. Now, eccentric contractions are a popular type of contraction to study because much of the muscle's normal activity occurs while it is actively lengthening. This is best described or illuminated when you think of your quadricep muscle that acts as a brake as you're descending down a staircase. So as you're walking downstairs, your quadricep is contracting, but it's allowing that muscle to lengthen to brake your momentum as you descend a staircase. Muscle injury and soreness are selectively associated with eccentric contractions, and so this is a very popular type of contraction to study in the experimental arena. An example of eccentric contractions is seen in a susceptibility to injury assay. In this assay, we have in this picture, let me show you here, this is an early picture from our laboratory. This is a mouse deeply anesthetized. The knee is affixed with a pin through the head of the tibia, and the foot is in the foot plate on the mounted to the torque sensor. And so the tibialis anterior muscle is being stimulated through the nerve with percutaneous stimulation. The knee appears surgically altered, but it is not. We just applied a depilatory to remove the hair from the skin of the animal. During the tetanic contraction, what you see is that the concentric phase, the isometric phase of this contraction starts. We hit a plateau period, and during that contraction, during the time of that contraction, I'm articulating this movie here. During the time of the contraction, the toes start to extend, the muscle contracts, and then we lengthen that foot. So the tibialis anterior muscle is stretched during the contraction phase. And on the right-hand side, you see that. You see the stimulation paradigm on the bottom, the stretch paradigm, or the movement of that torque sensor, where it says stretch, and above you see two eccentric contractions. What we have highlighted are the first contraction out of 20 and the 20th contraction. And what you can appreciate is after 20 contractions we have a force decrement, and this is consistent with the loss in muscle function due to those eccentric contractions. This is a common assay used in the muscular dystrophy field in that oh, I'm sorry, my animation altered. But what you can appreciate is the first contraction is seen in red, and the 20th contraction in the MDX is seen in gray. And so we have a significant drop in the contraction seen in the MDX compared to the wild type, which indicates the enhanced susceptibility to injury of that dystrophic muscle. This can be seen here in an aggregate plot of the data. On the y-axis, we have isometric force as percent of the initial contraction, and this is an isometric force taken at the peak of that plateau period prior to the eccentric stretch. And as you can appreciate, over time, the wild-type muscle doesn't have nearly as significant decrement as does the dystrophic skeletal muscle. The same paradigm can be applied with in vivo eccentric contractions, but instead of done eccentrically, can be done isometrically. This is just another example. It's actually in the same publication, in that these same contractions done isometrically result in a force loss, but to a less significant extent than those done eccentrically. And so the 1300A system, the 3-in-1 system, provides many options for functional assessment of neuromuscular phenotype in rodents. And while it is tempting to run all the assays possible on every animal that you might have, it's rarely feasible, it's not time efficient, or really cost effective. Each of you will be interested in a very specific assay, likely tailored to your specific interest, whether it be neuromuscular phenotyping, whether it be driving metabolism, whether it will be looking at pharmacological targets in your preparation. And so you've seen this graphic before. It's meant to highlight that there are many aspects within neuromuscular activation that define the overall neuromuscular phenotype. It's important to understand and appreciate the goal at which you're setting out to do these experiments. What the 300A allows is us to isolate experiments within that peripheral neuromuscular axis, such that we're taking away the motivational component and we're directly stimulating that motor nerve to directly isolate neuromuscular function in the periphery. The in vivo assays are moderate throughput. The assay is across the joint, which is the most physiologically relevant, and it is an assay that can be repeated over days or weeks, which is a possibility, which is nice for drug treatment studies, for studies of growth, for studies in aging models. The in-situ preparation is a more direct measure of specific muscle function, because the in-situ preparation, as you recall, is a partially surgically dissected preparation. The benefit is that the neurovascular bundle is maintained intact, and then, as we've seen, we can stimulate either the motor nerve to elicit the contraction or do some direct muscle stimulation to interrogate either aspect of the activation of the muscle. The in vitro preparation is a special preparation in that the muscle is taken out of the animal ex vivo, which allows a direct measure of muscle contractility independent of any of the neuronal integrity involved in that system. And as Ramsey also mentioned, it's a unique assay in that the muscle can be maintained ex vivo, and pharmacological measures can be applied that are directed directly to that muscle. And so function outputs we know would be directly attributed to that specific muscle type. This slide is to really highlight, however, that whatever your indication, whether it be in vivo, in situ, in vitro, there are a number of different aspects that you can tackle with your phenotyping. Really, your imagination is the limit. The 1300A system gives you a very nice platform to explore that in all of these assay types. So thank you very much, and I appreciate your time.
Thanks, Chris. That was great, and I've just taken over presentation controls, and at this point we're going to move right into our Q&A session. We've almost reached the full time for this webinar, so we're just going to extend it by a few minutes. If you can stay on with us, please do. If you have to run, not to worry, everything's being recorded and will be shared afterwards. So I'll also bring on Matt and Ramsey back online. Guys, are you with us?
Certainly
Yes.
Perfect. All right. Thanks for being here. And again, thank you for wonderful sections of your presentation as well. I'm going to get right into this Q&A. So first question, are certain muscles better suited to being tested with a certain technique? Chris, you were kind of on this point, so maybe you could continue, and Matt and Ramsey, you can chime in as well if you want to add.
Certainly, Andy. Yes, indeed, that's the case. The muscle size, what the muscle size limits is the ability for us to get nutrients to that muscle or drugs to that muscle if the muscle is taken ex vivo. So really the in vitro experiments are limited to small muscles, thin muscles, extensor digitorum longus, soleus, both of the mouse, strips of diaphragm, what have you, that can be well oxygenated. The larger muscles, tibialis anterior, gastrocnemius, what have you, they really need that neurovascular bundle to have the blood flow intact.
Okay. Yeah, any addition from Ramsey or Matt?
So keep in mind that in vivo you can also do certain muscles that otherwise would not be because of their geometry, such as the masseter or the forelimbs. But otherwise, yeah, there's certain muscle groups can only be done with a certain method only, while the EDL and soleus really can be used with any of the methods.
Perfect. Okay, thank you. Next question. Regarding the normalization with respect to cross-sectional area, Ramsey, you were mentioning this in your slides. What happens when the cross-sectional area is not uniform along the muscle? Or, is there a specific way to approach how to measure the cross-sectional area? How would that, can you elaborate on that?
So usually the cross-sectional area is not uniform across the muscle, and what we do is take the cross-sectional area at the center. The best approach is through histology and getting cross-sectional area of individual fibers in the belly of the muscle. Otherwise, muscle mass also provides a decent normalization. And I don't know if Chris agrees with me.
Oh,
but
yeah.
in general, it's by far the easiest, and the advantages of measuring cross-sectional area are only incremental.
I'll chime in on that. There are a number of ways to approach estimating measuring cross-section area. Obviously, histological measures, CT scans of the animal to measure cross-sectional area and also muscle mass. Certain muscles have certain muscle architectures. That results in muscles that have different what are called pennation angles, the directionality of how the muscles attach into the tendon. And there are some equations that are in the literature. There's a couple of key publications that you'll see routinely cited that are used to calculate cross-sectional area when across these different muscles. And so, for example, the extensor digitorum longus versus the soleus versus other muscles, the pennation angles are known, and this is a factor in an equation that can be used to calculate these. And so there's a number of those in the literature, and we'll make sure to put some of those key references in the reference list that gets put online.
Perfect. Okay, that's a great answer. Matt, we've had a few researchers with cardiovascular interests chime in, and just very simply, what would be the preparations and tests that are most common for those looking at cardiovascular muscle?
Well, I mean, one thing that I think is really beneficial, Andy, to look at that we didn't touch too much upon in this webinar is measuring work. that is something that that researchers with a cardiovascular bent would be interested in. And the way to assess work is basically to look at the area underneath the contraction performed. So if you're running a battery of contractions on a particular animal, in addition to those tests that we described, work is one that you would also want to consider, and that's of course something that is included in the software suite.
Perfect. Okay. And I'm assuming more information on those specific tests can be found, through your materials and website as well, for those that have a pure cardiac interest, correct?
Yeah, absolutely, and in the literature as well.
Okay.
And we have other specific chambers that are specific to isolated cardiac preparations as well. It's a little beyond the scope of this webinar, but we've done it for many years.
Perfect. Okay, coming back to muscle types, there's been a couple questions about identifying effects between slow versus fast twitch muscle fiber types. Can perhaps Chris Ramsey, can you take a lead on at a high level explaining how one would go about an assay designed to answer the difference between slow and fast twitch muscle?
Sure. In brief, the force frequency relationship is a good test to distinguish between fast versus slow muscle. Not only the peak force or the peak torque that's generated at each stimulation frequency is slightly different between the two. The shape of that force-frequency relationship is different between those two muscles. And that shape is based on both the excitability and the activation of those muscle cross bridges between the two muscles. And so if you did a force-frequency relationship between the soleus and a force-frequency relationship between EDL, you see distinct differences between those muscles. And where this is really helpful is if you have a disease or condition where there has been a significant fiber type change, exploring that the relative position or the relative slope of that force-frequency relationship is fairly indicative of the underlying differences in contractility governed by that fiber type change. And so this has been explored in many different publications from laboratories, including ours.
Excellent. Perfect. Well, in the interest of time, I'm going to say we'll end the live Q&A there. We've had a number of other questions come in from our audience, so thank you. These will all get answered, and transcribed response to this full Q&A session will be available for download. So let me officially thank our three presenters today and our sponsor, Aurora Scientific. Guys, you did a wonderful job. Thanks for being with us today.
Thanks everyone who joined.
Yeah, and a big thank you to our audience. For those that made it out to the live event, thank you. For your contributions and involvement today with our pollings. You're also going to be invited to complete a survey after logging out of today's webinar. Please take a couple minutes to tell us how you found the content and the delivery of today and what you'd like to see moving forward. This will help us identify new topics for the future. So thank you, everybody, and have a wonderful day.